Coral is not only an organism, it is an ecosystem, a host to a wide variety of organisms. It is what we call a symbiome. In order to better understand the coral symbiome, we use confocal microscopy, which allows us to observe the living coral in a non-invasive manner. Much can be learned from these observations, including the signature and distribution of innate fluorophores, the distribution and fluorescent emission of endosymbiotic algae, and the presence and characterization of coral larvae and associated epifauna. Therefore, we can look at the coral and all associated organisms as a whole and living entity. Here, only the innate fluorescence of the coral itself and its associated organisms is imaged. No dyes or fluorescent labels are used. The bright red coloring of this healthy coral is due to the fluorescence of chlorophyll contained in the endosymbiotic algae known as Symbiodinium. These unicellular organisms provide the coral with photosynthetic nutrients, and the coral in return provides them with necessary inorganic materials. The blue and green colors represent the coral's own fluorescent proteins. Time-lapse imaging shows us the movement of the polyps and the tissues that surround the polyps, known as the coenocerc. If you look closely, you can see tiny rotifers crawling on the surface of the coral. Confocal microscopy can give high-resolution images by eliminating light from outside of the focal plane. Layers of data from different focal planes, known as Z-stacks, can be collected and reassembled into three-dimensional images. Confocal images can clearly show the distribution of different fluorophores. Individual red dots of Symbiodinium can be seen in the tentacles and throughout the coenocerc. The fluorescent protein, represented by the blue color in this coral, seems to be closely associated with biological structures containing nematocysts, or stinging cells. Using the confocal microscope also allows us to see inside the coral, such as this view of the lace coral polyp's gastrovascular canal. A side view through this polyp's tentacles shows us the densely packed Symbiodinium and bands of tissue that bind the tentacles together.